When we consider how to increase the crop yields and the understanding of how plants control and direct the nutrients transport from the soils where they are produced into the sink tissues for growth and storage is fundamentally important. The plant has involved a very complex vascular system that can transport the sugars, proteins, and nutrients between their leaves, stems, roots, and fruits. This vascular system is not just a pipe for the bulk flow of the nutrients. It is able to actively direct the specific nutrients to specific tissues where and when they are needed. And my research has focused on the endpoint of the vascular system, where the nutrients are unloaded from the phloem into the growing part of the plants that need the nutrients. This endpoint involves the nutrients traveling through the nanochannels called plasmodesmata that link the plant cells. We identified a novel gene called phloem, which controls the outward trafficking of nutrients from the vascular system to cells. Phloem acts specifically at the interface between the phloem pericycle and endodermal cells. We found that the mutant plants that are missing the phloem genes has a higher rate of unloading, and as a consequence, their roots grow faster and longer.
So we wanted to understand how this increased permeability was being mediated in our mutant. So we decided to check a range of potential explanations. So we checked whether the density and the number of plasmodesmata was changed in our mutant, but it didn't seem to be the case. Then we checked whether there were changes in callus, which is a polysaccharide that lines the neck region of plasmodesmata, which is a known controller of permeability, but it didn't seem to be the case either. So then we decided to focus on the ultrastructure of plasmodesmata. And to do that, I went to Bordeaux to use electron tomography. So electron tomography is based on having your sample as a thin slice on a grid and then tilting this grid at different angles, and you acquire an image at each one of these angles, and then you can compile all these images into a single one, the tomogram. We could see that in terms of overall structure, so number of openings of your plasmodesmata, there were equal proportion of simple, one opening, and branched, multiple openings, plasmodesmata in both the mutant and wild type. But the real exciting difference in phloem was that we could no longer detect type 2 plasmodesmata. And what do I mean by type? Type is a new definition relating to how closely the plasma membrane and the ER membranes of plasmodesmata are. So for example, if we look over here, so in light blue you've got the ER, in yellow you've got your plasma membrane, and in the space in between you can see this red element over here that are tethering elements, the space the two membranes apart, so that you have a space in between the two called the cytoplasmic sleeve. So the canonical models of transport assign a key role to the cytoplasmic sleeve, the spacing between the plasma membrane and the ER. So the assumption was that the wider the cytoplasmic sleeve you had, the more conductive to transport you would be. What we actually find here is sort of the opposite. We actually see that type 1 plasmodesmata, those have closely opposed membranes and no obvious cytoplasmic sleeve, are actually more conductive to transport than the classical type 2 plasmodesmata, where there's the spacing in between the two.
Understanding how genes influence the transport of nutrients around the plants would be useful when we breed new crops. Ultimately, it might be possible to direct the transport of nutrients to specific tissues, such as seeds, fruits, and other storage tubers, which can increase crop yields. However, there are still many questions we have to answer before we reach that point, such as why the narrow plasmodesmata have a higher rate of trafficking, and how the metabolism of sphingolipids that we confirmed are linked to the function of phloem impact on the whole system.